Hello and welcome back. This is the video about antibodies. We could go into a lot of detail about the three-dimensional structure of an antibody, but we won't. I just want you to understand what they are, roughly how they are made, and what they do. So let's get into that. These are two diagrams of antibodies. This is the simplified diagram that helps you understand how they're constructed, and this is it's a snapshot of a three-dimensional model of the sort of space that would be filled by all the molecules, or all the atoms, I should say. Antibodies have three parts. I should also back up and say there are 5 different types of antibodies, but they all have some of these things in common. One thing they have is the antigen binding site, which we could also call the epitope binding site. This is where the epitope would attach. This is the incredibly specific part that only recognizes one epitope. And if you were to look over here, it's too complicated to even see. I wouldn't know where to begin to look for the antigen binding site. So antibodies are made up of 4 separate proteins, 2 identical what's called heavy chains, and they're just heavy because they're longer. There are more amino acids in these chains, so they're heavier. And then 2 light chains that are identical. Now, if you will recall, I told you that lymphocyte receptors are made by randomly shuffling DNA. And in this case, this part of the heavy chain is made by randomly shuffling DNA, and this part of the light chain is made by randomly shuffling DNA. So you get 2 randomly shuffled halves of the antigen binding site, and that exponentially increases the possibilities. So we have an astronomically huge number of possible antibodies a human body can make. Realistically, we don't make all of them. We randomly select the ones we can make, and we end up with a huge pool of possible ones. So that's kind of the review of where we've been so far. So let's get into this. What do? Well, they prevent virion attachment by sticking to the outside of a virion and blocking all of its spike proteins. And that's what would happen here. This would just attach to the spike protein, and this whole huge protein would be sticking off the side of the virus, and that really interferes with attachment. Because a spike protein is no bigger than this. So you have this whole thing sticking off the spike protein. There's no way the spike protein will interact with cell surface receptors the way it is supposed to. So antibodies can prevent virion attachment, and this is important. They can opsonize bacteria, or they can opsonize whatever they attach to. That's possible because in addition to this highly variable epitope binding site or antigen binding site, they have this not variable Fc fragment. This is not variable. That means that all the different antibodies and all their different antigen binding sites all have the same Fc fragment. And macrophages and dendritic cells and neutrophils all have receptors for the Fc fragment. And so if they ever see something with the Fc fragment on it, they know they need to eat that, because that means whatever it is coated with antibodies. Right, so this is what I said, okay. The Fc fragment, I just talked about it, it's an opsonin. Yeah, and the other thing it does is once the Fc fragment, or once an antibody binds something, the Fc fragment changes a little bit. And parts of the body can identify, oh, that's an Fc fragment from an antibody that has attached to something. One part of the body that can do that is the complement system in the blood. The complement system is a set of proteins in the blood that have some different functions. And one thing that triggers their functions is the Fc fragment of a bound antibody. If it's in the blood, it will trigger the complement system. So that's another thing antibodies can do. So here is going to be a list of what antibodies do. grouped into a few different groups. So if they attach to bacteria or viruses, they opsonize them. That's one thing they do. And that's a big deal, because that allows macrophages to attack and eat virions, even though they couldn't detect them with their toll-like receptors. Because the outside of a virion is going to be highly variable, and we don't have pattern recognition receptors for that. But we do have pattern recognition receptors, essentially, for our own antibodies. As I mentioned, if they attach to the outside of a virion, they block its interactions with cell surface receptors. But also, think about if they stick to a bacterium. If enough of them stick to a bacterium, let's go back and show you something. There are 2 binding sites on this one antibody. This can potentially attach 2 bacteria to each other. And so if they're trying to swim around and they're stuck together, they're not going to be able to move, or they're not going to be able to move in the way they want. So antibodies can do all these things to bacteria and viruses. They also protect us from protein toxins. So what's an example of a protein toxin? Well, the tetanus toxin and the botulism toxin are both proteins that have extreme effects on us, and they're incredibly toxic to us. There are a lot of proteins made by bacteria that are toxins, and bacteria use them when they cause diseases. They release them into us and they hurt us. The toxin that kills children who have diphtheria is another protein toxin. So how does our immune system protect us against those toxins? Well, antibodies attach to them. And antibodies attaching to a protein really interfere with its function. So all of these proteins need to interact with human cell surface receptors if they're going to change the behavior of our cells and hurt us that way. They can't do that if they're covered with antibodies. Also, proteins are much smaller than a virion or a bacterium. Those are huge compared to a single protein. But if you get a protein with a whole bunch of antibodies stuck to it, now it's big enough that it becomes an object that a macrophage can eat through phagocytosis. And then the last thing they can do is, like I mentioned, they can initiate the complement system, which is called the complement cascade, because it's a series of reactions that happens. Each one leads to the next. So this is a diagram that shows those effects. Everything except the very last one. So coating a virion, suddenly the spike proteins are not controlling what it does. Now it's got all these antibodies. This would be a protein toxin. It's just showing, it's not drawing the squiggle the way I would. It's just drawing it as a long wavy chain of amino acids. But with antibodies attached all over it, they will interfere with its ability to interact with our cells. And then a bacterium can be covered and opsonized. That's like, so can a virion. This would be opsonized as well as unable to attach. Agglutination and precipitation are similar to each other in that the antibodies are making a network of whatever they attach to, and that makes it act differently. So all these cells will suddenly act as one big blob of cells, or all these little proteins they've attached to suddenly act like a giant network of antibodies. And the end result of all of this is phagocytosis. So there you have it. This has been antibodies. In the next video, we will think about cells of the adaptive immunity, the education process, clonal expansion, and differentiation. So get ready for that. I'll see you there.